Okay, I hope all of you can see my screen.
Yeah, it's fine.
All right, so I'll be presenting part of the work which has been going on in the lab since last few months. Initially, there was so much difficulty in getting started sharing reagents that I had almost made up my mind that will not work on SARS-CoV-2, but it was blasphemous because we've been, at least I've been working throughout on emerging viruses. So we had to do something, but then we got hold of a few reagents which allowed us to do a few things, and one of those projects I'll share the data on. This is about all the strategies SARS-CoV-2 has against the cellular innate immune response. So this is a SARS-CoV-2 genome and proteome. All of you have seen. I'll point out a few things which are not obvious. One, it has a lot of proteins being made, either as polyprotein being processed into smaller proteins, a lot of different strategies, ribosomal frame shift, bunch of small genomic RNAs. In the end, it's making close to 30 proteins. New data suggests that there might be even more number of proteins which are not clearly described yet. But if one tries to look at these proteins and understand what is it that virus really making, what kind of proteins are being made. So we see pictures of the virus all the time, but if you look at that picture, it's mostly made of the structural proteins, which are spike, envelope matrix, and nucleoprotein, and they contribute or they come from a very small part of the virus genome. So what is the rest of the virus genome dedicated to? So obviously viruses have very tight limitation on the size of the genome they can package in their body or the virion. This virus in particular has about 3 times larger genome compared to any other RNA virus. But still, the structural proteins, which one would think are the most important for virus infection, they contribute to a very small proportion of its genome. Most of the proteins have other functions. So what are those proteins? Then there is a set of proteins called non-structural proteins. About 16 of them are made. Some of them are understood that they have the polymerase, the RNA-dependent RNA polymerase activity or helicase activity, so may be involved in making more copies of the genome, but many of them are there whose functions are not known. And then these accessory proteins, which are made from subgenomic RNAs. So why it is important for virus to carry a sizable part of its genome, which will make these accessory proteins. And if you just look at size of the genome required for it, it's almost equivalent to what is making the structural proteins. So obviously it is doing something which is very important for the virus replication. So what is mostly done by these proteins, either they are completely dedicated or they are at least moonlighting to counter the cellular innate immune response, which happens this way as shown here. And this is probably much more fight which virus has to fight inside the cell before even it is targeted by adaptive immunity, both B and T cell. And if you look at the number of proteins it encodes, the amount of genome which dedicates to these functions, it becomes clear that this fight which goes on between the viral proteins and host proteins inside the cell is very, very critical for viral survival. So put very simply, what is shown here is that upon SARS-CoV infection in any cell type, say an epithelial cell in our trachea or lung, it will be detected, its genome will be detected. That is done usually by a Rigi-like receptor, and then what ensues is called RLR signaling or Rigi-like receptor signaling. The signal is conveyed to an adapter molecule on the mitochondrial membrane. This in turn will activate a series of kinases which will phosphorylate these set of transcription factors. The one shown here is IRF3, but NF-kappa B and IRF7 also contribute. Once phosphorylated, they will move inside the nucleus and they will bind to the promoter regions which are driving expression of what are called interferons. Now, interferons, there are a bunch of them. There are 3 types, and then in type 1 there are about a dozen of them. But in general, all of them, they go towards a similar line that they activate the neighboring cell, or they come back on the same cell in autocrine manner, and then they initiate this pathway called JAK-STAT pathway. which will lead to activation of STATs, the STAT transcription factor. Once they move inside the nucleus, they will drive expression of what are called IISGs, or interferon stimulated genes, which can range from anywhere from 400 up to 1000. And they are the ones which are the fighters, the communicators, which prime the adaptive immunity, which fight the incoming virus. Now, the interferon induction part is distinct, called interferon induction here, and subsequent part is called interferon signaling. And all these distinct steps, they present an opportunity for the viruses to disrupt these things so that IISG production can be mitigated or prevented completely. So this is what we wanted to study in context of SARS-CoV-2, that what SARS-CoV-2 proteins interfere with these processes. And for that, we started by contacting our previous collaborator, Professor Krogan in University of California, San Francisco. And they very graciously provided us the whole battery of expression constructs of SARS-CoV-2. Few proteins were missing, but most of them are there. And they had published a protein interactome map of all these proteins, SARS-CoV-2 proteins. We used these constructs, we tested them for their expression by Western. These all proteins are tagged with a struct tag. So you can see all the non-structural proteins, some structural proteins, and a bunch of accessory proteins were there. Then we started testing them for their ability to inhibit either interferon induction or subsequent interferon signaling. So this is done by a luciferase-based assay, where luciferase is either being driven by interferon beta promoter, or it's being driven by an ISRE promoter, which is required for ISG induction. So if you look at structural proteins and their ability to interfere with these processes as compared to a control vector, there is not much. Actually, you see a little bit of upregulation, but in general, they do not interfere with these processes. So they have other things to deal with. They are directly targeted by the adaptive immunity, and they are not burdened by the virus to deal with this intrinsic immunity or innate immunity. Then next we look at all the non-structural proteins from NSP1 to 15. And here what you can see is that there are few proteins which can clearly shut down the interferon induction. Also subsequently, the ISG induction also is inhibited by many of them. One clear hit here is the NSP1 protein, which was reported in an article in Science, that in general it just shuts down translation, it interferes with the ribosomes. Then there are proteins such as NSP13 and 14, which are also very effective in inhibiting both interferon induction and ISG signaling. NSP5 is a protease that is also seen to having some inhibitory effect. Next, we will look at accessory proteins. Here also, you can see that many of them, especially ORF6 and ORF9b, are very effective in inhibiting interferon induction. And for ISG induction, there are more proteins which are having inhibitory effect. So here's the summary of all of that. This column shows inhibition of interferon induction, this part. And this column shows inhibition of ISG, or interferon stimulated antiviral gene induction. And these are all the proteins categorized in structural, non-structural, or accessory. So you can see structural proteins are not bothered by it. They have other things to deal with. The responsibility of dealing with cellular innate immunity is shared by non-structural and ORF6 proteins. Now, these non-structural proteins, as I mentioned, although they do not constitute the virus body itself, but they make some sort of enzymes which are required for, say, viral genome replication. So if one has to knock them out in order to mediate attenuation of the virus, that may not be possible. Whereas these accessory proteins, they appear and disappear between closely related viruses. So these are sort of dispensable. So that's why we decided to focus on this protein called ORF6, which was showing most potent activity in inhibiting both interferon induction and ISG induction. And here's the summary that these are the proteins which are inhibiting interferon production, and these are the proteins from SARS-CoV-2 which are inhibiting subsequent signaling. But at what step they are interfering, those things were not clear. Since we started working on it, close to a dozen papers have come out. Many of these proteins' role have been addressed. A lot of them remain addressed, so we'll work on that. But for now, I'm going to share data which we got with ORF6. So very interesting thing about ORF6 is that it appeared in 2003 SARS-CoV virus, and it is still present in current SARS-CoV-2 virus. But if you look in other closely related human coronaviruses, say seasonal coronavirus or MERS virus, it is not found there. So it is a dispensable protein to say. Now it was present in SARS-CoV, present in current coronavirus. It does not mean it's the same protein. A similar protein is present, which has some sequence differences. So first of all, you wanted to see in the interferon induction pathway where all R6 could be interfering. So we started by testing at different steps. So first of all, we did the interferon beta luciferase assay, where the interferon beta luciferase was activated by what is called 2-CAR domain, which is a part of this regi, which is directly responsible for activation of the pathway. So in case of vector, you can get 100% induction in presence of 2-CAR, but if you co-transfect OFF6, that induction is completely inhibited. Next step would be TBK1, which is a kinase required for phosphorylation of IRF3. If you overexpress TBK1, you get a nice induction, and if you co-express OFF6, you get inhibition of TBK1 also. Further on, we tested IRF3. We used a molecule called IRF3-5D, which has 5 aspartic acid changes, and that's why this is a phosphomimetic. So this acts like a constitutively active IRF3. And when you express this into cells, interferon beta induction is high. But if you co-express OFF6, this is also inhibited. So it was clear that OFF6 was probably acting very much upstream here at the level of regi, and that was leading to subsequent inhibition of all these steps. Or maybe it is possible that it is independently inhibiting these steps also. The immunofluorescence here shows inhibition of nuclear localization of IRF3 upon expression of OFF6. So these cells are expressing OFF6, the neighboring cells are not expressing. And where OFF6 is being expressed, you can see the IRF3 is mostly cytoplasmic, but in a neighboring cell where OFF6 is not there, IRF3 is in nucleus. And this is in presence of an stimulus, which will allow IRF3 activation and movement inside the nucleus. So this is how we have figured out so far that OFF6 might be interfering with interferon induction. But remember, it was also inhibiting the downstream interferon signaling, which might result just by the virtue of inhibiting interferon induction also, but it may have independent mechanisms as well. So it was reported in case of 2003 SARS-CoV that the R6 protein present there was targeting STAT1. So first of all, we tested whether or not R6 of SARS-CoV-2 interacts with STAT1 and STAT2. So we did a co-immunoprecision expression, and we found that only when you have R6 and STAT2 present together, you can pull down R6. With STAT1, you're not able to pull down R6, which means that R6 interacts exclusively with STAT2. Here is the input data related to that co-IP. And if you do immunofluorescence, you can see that R6, it sort of sits around perinuclear region. Now new reports have come which shows that it is actually inserted in the nuclear membrane. And while sitting there, it pretty much blocks nuclear entry of all these key transcription factors which are required for interferon induction and signaling. So in interferon signaling, it is inhibiting STAT movement inside the nucleus. So it has multiple ways by which it is inhibiting the antiviral innate response inside the cells. Now, if we look at the SARS-CoV-2 R6 protein sequence, as I mentioned, this was not present in other human coronaviruses, both seasonal or MERS, only present in the original SARS and SARS-CoV-2. But if you look at other closely related sequences from other animals, what we see here is that R6, which SARS-CoV-2 has, it resembles the one which is present in the bat coronavirus, which is supposed to be the progenitor of the SARS-CoV-2 strain itself. And next closest relative is the pangolin virus. So it pretty much follows the pattern followed by the entire genome comparison also. But if you look at the sequence alignment, these stars represent the conserved sequences. It clearly shows that although they have the same protein, the sequences are not identical. Sequences are quite different. Also, it was reported, at least based on modeling, that the structure of SARS-CoV is such. It has a N-terminus cytoplasmic region, a C-terminus cytoplasmic tail, and a transmembrane region. So this transmembrane region, which is a little long, this has a lot of these conserved residues. So this got us interested into it. So we thought we'll map it and try to see what effect it has or what role it has in inhibiting interferon induction and signaling. So we made these alanine scanning mutations at the conserved sites. In 4 amino acids each, we changed all these conserved residues and made mutants 1, 2, and 3. And then we are using these mutants in comparison with the wild type OF6 to see whether or not their ability to inhibit interferon induction or IISc induction is affected. So it shows that the OF6M1, it has got a hit, its ability to inhibit interferon induction was affected. As you go more towards the end of the protein, it's not affected. Same applies to IISc induction also, but remember these two functions are independent. This is happening probably by targeting STAT2, and this is happening probably by targeting RIG-I or something from RIG-I to TVK to IRF3. So which means that these residues are probably more important for maybe interaction with those components in interferon induction. And this membrane proximal region here is maybe critical for that. So this is what we have in terms of data so far. What we are doing right now is trying to understand the mechanism of inhibition of interferon induction by SARS-CoV-2 a little bit more detail. And we are also working on developing a SARS-CoV-2 reverse genetic system. The aim would be to see if these accessory proteins, which are very critical for interferon inhibition, if they can be knocked out of the SARS-CoV-2 genome, will it become highly attenuated? And in addition, we are looking at other SARS-CoV-2 proteins also, which were described earlier, how they are interfering with interferon signaling and induction. And all of this work has been done by really hardworking and talented students in the lab, Mansi, Vaida, and Rohan, who's a postdoc in the lab. They have really braved the pandemic, worked day in, day out, faced weeks-long quarantine to do all this work. So credit goes to them. The reagents, especially the SARS-CoV protein expression constructs and the plasmids required to study interferon signaling, these were obtained from Naman Krogan and Adolfo Garcia Sastre, which are previous collaborators. And financial support for this particular work was provided by IISc as MHRD funds, the startup funds which I received when I joined the institute, also by Infosys Foundation in form of Infosys Young Investigator Award. And Rohan, who worked on the project, he's a postdoc and he's supported by India Alliance DBT Welcome Intermediate Fellowship. And that's all I had. I'll take questions if there are.
Okay, great. Thank you. Now I'm getting. Are you able to hear me?
Yes, I'm hearing you.
Okay, so there are a few questions. One is from Manapurna. How much evidence is there that structural genes which remain IFN inducing are accumulating mutations that move them to non-structural proteins which can inhibit induction? And secondly, is there any evidence of selection pressure on the virus in viral strains circulating in India? So I didn't understand the first question. But,
me neither. So
maybe Anupurna can open up her mic and ask it.
Yeah, Shashank, I mean, what you're showing is that the non-structural proteins are able to inhibit interferon induction, whereas the structural proteins are not. So the question is there any evidence of selection pressure on the virus in strains circulating in India that more and more genes become inhibitory rather than remain capable of inducing? Is there any evidence of the Genome Project highlighting mutations which make them less capable of inducing interferon? Are there in specific genes which are capable of impacting interferon induction? Do you know of any? Is there selection pressure?
Well, I've not looked closely at the sequence data, especially from India, but one thing which was mentioned is quite obvious, that this protein was absent in seasonal human coronaviruses. It's just picked up by these more pathogenic SARS-CoVs. And then again, it's absent in MERS, so whether or not it directly plays a role in viral pathogenesis, we don't know, because MERS is the most pathogenic coronaviruses currently known. So I have not looked closely at the interferon antagonists, but in general, the proteins which play interferon antagonistic role in viruses, they are under selection pressure all the time. This has been seen in flu, in flaviviruses. So the regions which code for those non-structural or accessory proteins which have interferon antagonistic activity, those are actually most flexible regions. Those are the regions which can be mutated, can be removed, and also changing very rapidly. Yeah.
No, thank you. Thank you, Shashank. I also had just a follow-up question on whether you want to comment on the presence of these rare auto anti-interferon antibodies that have been found in patients with severe SARS-CoV-2 infection. Any thoughts on that in the context is that it has emerged in quite a few countries that patients with severe CoV-2, a small proportion of them have these auto anti-interferon antibodies. Any evidence in India in the samples that you have screened for evidence of this?
Okay, no. Quick answer, Shashank. Okay.
No, I'm not familiar with that. No, sorry, no. Okay. That's a new thing. Yeah.
Yeah, question from Narendra. Are there IIScs that block ORF functioning, ORF6 functioning?
Yeah, so ORF6 function other than interferon antagonism, it's not clear either. So if we know its otherwise function, then we would know if that is being inhibited by cellular proteins or not. Like I said, we still are trying to figure out what are the different mechanisms by which it is targeting interferon induction and signaling. So proteins which are usually targeted by antiviral factors are things which are required for virus entry, budding assembly, or viral genome replications, which are easier to understand actually. These accessory proteins, other than interferon antagonism, other roles are pretty vague. It's hard to study.
Okay, Kumar has a question. Is the different size ORF3A protein in SARS-CoV-2 responsible for reduced interferon response?
Yeah, so there was a paper on that from a Japanese group. In our hands, we didn't see much antagonistic activity with ORF3 proteins, although it was reported in case of original SARS also. In our hands, among ORFs, we saw activity with ORF6 and ORF9b only. With ORF3 we did not see, but there is a cell reports paper which shows that ORF3A, 3B also has activity against interferon.
Shashank, this was based on comparison of ORF3A from SARS-CoV-1, which had a much bigger ORF3A.
Correct. Yeah, I'm familiar with that study, but what I'm saying is that we also tested ORF3 constructs from SARS-CoV-2. We did not see interferon antagonistic activity. So these items they become consolidated when multiple papers report the same thing again and again. So this is a still emerging thing. When consensus develops, we'll know which proteins are more important. All right.
Okay, Shashank. Thank you.
Yeah. Thanks a lot, Arpan. Thanks for